Let's welcome Mr. Alex Olengo. He's here behind me. People are supposed to clap for you as you come up, but let's welcome him. Thank you so much.
Thank you very much. Once again, by the names of Alex Olengo, and I'll be the chair for this session. I think it's my presentation that is actually coming next. The chair and the presenter, Arego. This is basically work that I did for my master thesis, titled Molecular Cloning and Recombinant Protein Expression of 9 LOX, or 9 lipoxygenase gene from solanum tuberosum in pichia pastoris yeast cells. So it's just basically the presentation outline, how it will flow. And of course, the abbreviations that we shall meet, I'll be using 9 LOX gene for 9 lipoxygenase. So as a preamble, the lipoxygenases are actually ubiquitous in both plants and animals. They are found in both plants and animals, and their role is basically to facilitate the addition of molecular oxygen into polyunsaturated fatty acids in order to form hydroperoxides. And of course, in the biosynthetic pathway, the hydroperoxides are transformed into oxalipins that confer antimicrobial resistance, and that is one of the reasons as to why we went into this kind of research. So the problem that we had here was that synthetic fungicides have led to disruption of natural biological systems and also sometimes fungal resistance, and they also have undesirable effects on non-target organisms. So hence the view to go into research into biodegradable fungicides that do not have bioaccumulation. So the justification, the study has proven that pichia pastoris can generate a recombinant 9 LOX protein. And of course, we generated cell lines that could be scaled up for the lab production or chemoenzymatic synthesis of the oxalipin-based fungicides. So the general objective of the study was to produce recombinant solanum tuberosum 9 LOX protein using pichia pastoris. Solanum tuberosum is an Irish potato. Yeah, and it's the one that contains this enzyme. Tomatoes also contain the same enzymes, and of course cereals as well. So the specific objective was to generate recombinant PPIC-Z alpha B plasmid vector. This is it's a eukaryotic vector that is compatible with pichia. And we used, to generate the recombinant plasmid, of course we used golden gate cloning. And then the second objective was to generate recombinant 9 LOX protein. That is protein expression, basically. So the flow of the work was basically cloning the 9 LOX gene into PPIC vector and then transforming it to generate more copies of the recombinant vector, then purifying it and then transforming it in pichia pastoris using electroporation method, and then after expression of the recombinant protein, and then downstream the analysis using dot blot, SDS page, that is sodium dodecyl sulfate, polyacrylamide gel electrophoresis, and then Western blot analysis, and of course data interpretation and presentation. So briefly, this is what we use the NTI vector program to cut the gene into 3 pieces and then subclone these segments or these gene fragments into what we call level 0 intermediary vectors to hold the pieces of the genes in position. The reason as to why we did not do restriction ligation cloning was because of the large size of the gene, which could not be cloned directly into that destination vector or PPIC-Z alpha B. So that's why we had to cut the genes into pieces, subclone them into intermediary vectors. Then also we needed to prepare the destination vector. You know, the vector PPIC-Z alpha B comes with a selection marker called zeocin. And at the end of the day, after transforming, you have all white colonies. So it's difficult to select which one is a transformat, which one is not. But if we have this blue-white selection, it's a bit easy for us, and that's what we did. So after preparing the destination vector, then the small pieces of the genes, we went into the Golden Gate reaction, where we have equimolar amounts of the specific intermediary vectors and then the destination vector in one PCR tube. And then we add a type 2 restriction enzyme, which is actually non-palindromic. Once it cuts, there is no more cutting. So there is what we call seamless joining of these genes without any error. Actually, this kind of cloning has an efficiency of around 95%. So that is basically what we did after sequencing. This work was done in Germany, but then the expression work was done from Makerere University College of Vet Medicine. So remember, the pichia genome is actually linear. So after having a recombinant PPIC Z alpha B containing the full cDNA or the full 9 LOX gene, you have to actually linearize it using a PME enzyme in order to aid for homologous recombination into the pichia genome. Thereafter, you'll need to transform and, of course, select your colonies, and then you start protein expression in baffled flasks. That's for pichia. After expression, like I told you, we do the analysis, dot blot analysis, SDS, and of course Western blot. Perfect. Just to flash on the results, these are amplicons of 9 LOX gene fragments. That is the LAC-Z fragment that we amplified from another vector in order to introduce it into our destination vector PPIC, Z alpha B. And of course, the final product, we did restriction enzyme analysis on the final product and everything was fine, even after we went ahead and sequenced. So, that is a PME1 enzyme, a representative gel that shows linearization, linearized and unlinearized recombinant plasmid vector. So this is a dot blot analysis that actually showed expression of the protein from the cell lysate. In our discussion, I think there is one of the slides that I missed out that had a slide that had the SDS page and of course the Western blot. But it showed a putative band size of 97 kDa, which is the theoretical band size of 9 LOX. So within the discussion, of course, the study was able to clone the 2.6 kb 9 LOX gene. And of course, all the principles of Golden Gate cloning were followed and adhered to. SDS page analysis gave a 97 kDa band, like I said. So in conclusion, the production of this protein from solanum was recovered from cell lysates using pichia pastoris X33 strain. The recombination, the recommendations progress made so far is the optimization of the alpha secretion factor, which is actually a gene within the vector, within the destination vector that directs synthesis of this protein into the media. But in this case, in our case, we found our protein within the cells. They were trapped within the cells. So we also advise artificial addition of the KEX2 protease gene into the host genomic DNA. This KEX2 actually is the one that cleaves the protein and releases it into the media. So that's the acknowledgement that I have from my supervisors and advisors. And of course, besides the ones I worked with, I'm happy to acknowledge synthetic biology as well. They are here to stay. And of course, we have a story to tell with them, a story of autonomy and purpose. Thank you very much for listening.